Good morning. My name is Elena Newell. I am an assistant professor at Baylor College of Medicine in the Center for Educational Outreach. What we do in the center is it's pretty multifaceted. Actually, we do a lot of teacher professional development. We do a lot of curriculum development, and we have affiliate middle schools. BCMA at Ryan and BCMA-R at Rusk, as well as DeBakey High School for Health Professions. I am an internal evaluator, and so in addition to helping out with professional development and curriculum development, my main role is getting evaluation going. So I work with faculty as well as students and across the educational spectrum. So I'm working with faculty at the Baylor and at the medical school, and I'm working with teachers who are doing professional development for kindergartners. So I'm all over the place. Today we're going to be talking a little bit about something that I see happening on a regular basis with our faculty and with teachers across the levels as they're trying to get research going. There's an issue with figuring out, okay, how do I analyze this? How do I get started? And so today is a very brief intro. I mean, this could be a master's course. So it's a very, very brief intro to data analysis and visualization, and specifically for helping students get going with science fair projects. So I will go ahead and move forward. Okay, so this is kind of our overview of what we're going to do today. So the road to results, if you're trying to get to results from a science fair project, where do you start? How do you get going? And then steps basically in quantitative analysis, which, qualitative analysis is wonderful and should be used and has its own steps. Today, we're going to focus on quantitative, but I do encourage those of you who might be interested in using qualitative measures to do some research into those methodologies as well, because they are very, very useful. And then we'll share some resources. And please feel free, if you have any questions or any confusion or any thoughts that you want to share as we are going, please pop them in the chat or raise a hand to let me know, and I would be happy to answer them as we go. So this is a kind of a typical approach to research, right? Where you pick out a topic, you read some literature, figure out what's out there, you develop questions, and then you decide how you're gonna study it, right? You come up with procedures, you collect your data, and then you get to your analysis. And that's usually towards the end of the road, as you might say. It's something that people think about at the end. The problem is that you don't want to find out when you get there that the data that you've collected is incorrect, or it's not going to help you answer your question. So really, the time to think about analysis is not all the way at the end after you've collected the data, but it's at every point along the way. It's part of the trip, as you would say, part of the journey. So take a moment. Think about a science fair project question, or one that you've used in the past, one that you're kind of wondering about, one that you've seen a student use or a peer, and just go ahead and share in the chat. And as you're thinking about it, also make sure that you write it down, because it doesn't have to be perfect, but it's kind of helpful to, as we walk through this process, to have an idea about your own approach or how you might apply it in your own work. So go ahead, drop some suggestions in the chat for science fair projects that you've seen or that you're interested in looking at, and we can go forward, maybe using some of those examples. Okay, comparison of rocket fin shape to maximize height reached. That sounds fun. Percent of vitamin C in orange juice. Well, that's interesting. So I imagine you could look at different kinds of orange juice, or like homemade fresh squeezed versus what you find in different store brands. And again, these don't have to be perfect. They're just kind of ideas to get going, because it's useful to walk through this with an idea kind of in your head for something that you might look at as well. So even if you don't feel comfortable sharing in the chat, please do write something down or kind of think about ideas that you might have on your own. The effectiveness of disinfectants. That is a very relevant one right now. That's one I think that scientists at all levels are looking at. Identifying variables to improve solar-powered RC car speed. Oh, that's awesome. Super fun. Okay, love these. So thinking about this, with your question, whether you shared it in the chat or whether it's just something you wrote down on your own, identify the variables. And this is important because I find that with a lot of people in general, not just students, but sometimes with faculty, identifying our variables and operationalizing our questions in a way that we can say, okay, I'm measuring this, this, and this at this time is a big part of getting to collecting the data that you need for an appropriate analysis. So go ahead, you don't have to share it in the chat, but do write down your variables and think to yourself, okay, can I identify what the variables are? And one of our chat suggestions we had, it's definitely about variables. They've already got some of these. They've got, you know, speed, amount of sunlight, wheels, so they've identified things. But think about your question and what you might be measuring. And this is important because and I will talk about it going forward, but this is important because if you can't identify that, then sometimes you might not collect the data that you need to answer the question. So we'll keep going forward. This is an example that I came up with, and this is actually from a science fair project that I've seen before, and this was at the lower middle school level. It's like sixth grade. And when you're thinking about a question like this, what you want to be able to see is the things to measure, right? So you look at this question and you say, okay, well, my independent variable is light, and I've got my dependent variable, something about growth, right? And you should be able to, with any kind of question that you pull out, identify immediately off the bat the independent variable and the dependent variable. And in this case, I've also called out the which, because that tells me that there's going to be a comparison, and the time, the rate of time, fastest, because that tells me something else about how I'm going to be measuring things. And so typically what I suggest that people do when they're trying to go from this to a question to data is to really detail it out more. And it's one of those things that I'm sure those of you who have dealt, especially with like little kids, you'll find, you know, that situation where you've asked a question, you don't get quite the answer that you're looking for because of the way you phrased it. You might say something like, what happened to the lamp? And the answer you might get is, I didn't do it. And that's not, I mean, it tells you what didn't happen to the lamp, but it doesn't tell you anything about what actually happened. So then you find yourself rephrasing and saying, how exactly did the lamp end up on the floor broken into pieces? And you provide more detail in the question so that the answer you get is more precise. So in this case, with this question, I made it more precise. So instead of being, okay, what color, we say, okay, which of the 3 light colors, and you could even detail, you know, red, green, or blue, makes plant height. So we're talking about growth in height, not width or number of leaves or thickness of the trunk. What makes plant height increase the fastest over a 6-week time period. So now you know exactly what you're changing, your independent variable, you know what you're measuring, so what your dependent variable is that you're looking at to change, and the time period that you're looking at it in. So there's some amount of time over the 6-week time period that you're looking at plant height. And that's really, really helpful because it means that when you go to answer your question, you're getting the data that you need. So part of this is all because the question that you should revisit multiple times during this process is, how will I know when I have the answer to my question? And I'm going to give you an example using the same example to kind of show you what I mean. So before we get to that, take a moment and think about the question that you've written down and what an answer might look like. And not the answer that like you're hoping for, like I'm hoping this shows that, you know, that the amount of sunlight is the best for this kind of car. What you're looking at is, okay, what would tell me that I have an answer? Whether it's the answer that I'm kind of hoping for or a completely surprise answer, what tells me that I have an answer? Because looking at the question that I've come up with, you know, I can think about an answer in this way. Okay, plants exposed to the red light grew the fastest. And someone might say, well, what do you mean by fastest? And so I might say, okay, well, plants exposed to the red light grew the fastest because they increased in height an average of 1.5 centimeters each day. And I can talk about the other kinds of plants and how their height grew. And so in this case, I've got data that allows me to answer my question with precision. Let's say that instead of measuring at multiple points in time, instead I just measured at the end. And so I'm going to say, oh, Well, the plants exposed to the red light grew the fastest. They were 19 centimeters tall on average at the end of 6 weeks, while the plants in the other groups were not as tall. The problem with this is, okay, they're similar, right? Like they seem really the same in a lot of ways. But what if the lights in the blue plant just exploded? So they went much faster. They were, 10 centimeters tall at week 2, whereas the red plants were just kind of eking along and they were much shorter, and they just topped out at a shorter height. Well, if that's the case, then what I'm measuring is not the speed that they changed. I'm not looking at the fastest. I'm actually getting which plants ended up being the tallest at the end, which might be something different. If I want my plants to grow really quickly, then I want to know the rate over time. If I just want them to be the tallest, then I just care about the end. And so when you're planning your data collection and you're thinking about your questions, little differences in precision makes a big difference in how you collect your data, and it makes a big difference in the end on how you analyze this data. So, okay, we've got our question, we're on the road. I know what I'm gonna measure, I know how many measurements I need, I've determined that I'm looking at fastest, not total height. Where do we go from here? So this is where we're gonna jump into not quite analysis yet. We're gonna look at the data that we're collecting. And part of that is that your analysis cannot make up for it if you're not collecting the right data. So we've got our measurement points, but it's really important to think about the nature of the data that you have. So one of those things is scale of measurement, which is a mistake that I think a lot of people make, is not picking the right scale. And part of that goes into what you can do with the data at the analysis point to answer your question. So the scales of measurement, and a lot of you might be familiar with this, there's 4 main ones that we really think about. And the first is nominal, where you use a number, but it's not not as a measure. You could replace it with pictures or any other kind of identifier. In this case, Altuve is not 27th of anything. That's just the number that he has on his jersey. Tell us who he is. It's an identifier. So in this case, if you use these identifying numbers, it wouldn't make sense to do something like take an average, right? Because they're meaningless in that way. So when I'm talking about analyses, you can kind of see where the scale influences that. Ordinal, we see a lot. So, of course, Olympic medals are an easy one because they're ranked. There's an order to them. But if you're a 1/10 of a second apart between gold and silver, we heard that a lot earlier this summer, right? Like, they were just, a 1/10 of a second off the medal stand. Well, it doesn't tell you whether that's 10 minutes or a 1/10 of a second. It doesn't matter. In this case, they're ranked. And the problem with this comes in is when a lot of surveys, you'll see things like, on a scale of 1 to 5, tell me how excited you are to be here today. Well, what's the difference between a 4 and a 5? What does that mean in terms of excitement? So if we take these numbers and we pretend like my 4 and your 4 are the same, and the same difference between a 4 and the 5 exists, and we take an average, what is that really telling us? Those numbers don't have an interpretable difference between them. So it's something you have to be cautious with those kinds of scales. Now, there are surveys out there where what they do is they aggregate multiple items in a survey all together, and then they can use those numbers to do things like averages. But in just one question on its own, what does a 3.8 mean if you take an average of satisfaction? It might not be telling you what you need to know. So then we move to interval, which temperature is a common one. It's the values in between, the distance between each value they're equal, but there's ratios won't make sense with these numbers. You don't get twice as burned if, the oven is 350 degrees as you would from 175. It's just, that's not how that works. And then final, ratio, which probably is one of the more common because there's a true zero. So any count data, height data, anything like that is going to end up being ratio. So that's what you can do. You can do a lot with ratio data, which is, super fun, but it's important to think about also because if you're measuring something like growing the fastest, it makes more sense to measure every day with ratio data. So meaning, the absolute, like the number, the height of each plant each day, versus if you just rank the plants, okay, which one is the highest today? That doesn't tell you as much, and you can't do much with it analytically as you could if you had ratio data. So that's why these scales are important. And so again, just kind of to reiterate, just as a summary, this is what will help you figure out the kinds of data that you need in order to make sure that you're answering your question. So sample size is another thing, and sample size is important on a couple different levels. Before I get into it though, I would like your thoughts. For you guys, why does sample size matter? Why does it matter how many repetitions you have in an experiment or how many people you ask to take a survey? And you can just drop it in the chat. And you don't necessarily have to turn on your camera and talk, but let us know. What are your thoughts on why sample size matters? Precision, yes. Any other thoughts? Okay, so I will jump ahead. Oh, more samples, more chance of getting a true answer. Yeah, that's exactly it. So when you're thinking about sample size, if you say small sample sizes aren't able to discern statistically viable differences, yeah. Confidence intervals, yes. Okay, so you guys are really there on sample size. Awesome. Thank you. So the thing with sample sizes is that, for those of you who aren't as familiar with thinking about why they're important, if you say, I want to know how fast football players are, and you grab 10 football players from all across the country at every level, but 2 of your players happen to be NFL players, and they're really, really fast, then you might think, oh my gosh, the average football player is way faster than most other people. Well, you've got 2 out of that 10, so 20% of those are all, skewed data bias. So it's completely biased, right? Because you've got these super fast people that are throwing off your scale. So if you want to be able to look at your results and generalize, you have to have a sample size that's sufficient to give you the information that you need. In this case, for a science fair project, there's other, for research in general, there's other considerations to take into account. So there's practical, like you would love to be able to survey every football player in the entire country, but that's really hard to do. You just don't have time and resources. Ethically, especially if there's living things involved, whether it's a person doing a survey or whether it's, like biological, biomedical research that you do a lot of at our labs here at Baylor, it's just not ethical to do more than you need. And then, analysis, as been pointed out in the chat, some statistical approaches will not work appropriately. They will not give you answers that are viable, that you can really depend on if you don't have enough of the sample. They can get really skewed by outliers, and they can be really impacted by weird quirks in a small population. So it's like if you go into a classroom and you survey all of the girls, you might get a different perception about what, satisfaction or how things are going, classroom atmosphere, than you might if you survey all of the boys. You want a representative sample. So these are things to think of in your research as you're going forward. So deciding on sample size is complicated, and it depends on the analysis that you're using. For younger scientists who are looking at kind of just getting started, 5 to 10 trials of something. So how many pea plants are, how many flowers come on a pea plant? Well, you might want to have 5 to 10 plants, just to give them an idea of flavor of the data. So the really little kids. For older scientists, so our learners that are in later elementary, moving into middle and high school, a rule of thumb, even if it's descriptive and you're not going to get into inferential statistics, is 25 to 30 trials per event, per category. So in my example with the light colors, then I would want to have 25 to 30 plants in each of the colors, so that I could measure those. Just to kind of make sure that I've got sufficient, and again, this is a rule of thumb, it depends on the kinds of analyses that I would be doing with it, but just as kind of I don't really know, how do we get started? For students who are using inferential statistics, there are calculators online that they can use to figure out what they're going to do. And we'll talk a little bit about inferential statistics as I go on, but just kind of as an idea, there are calculators that can help with figuring out the sample size needed. So we're going to go into quantitative data analysis steps. So finally, we're at analysis. After all this talk about kind of preparing yourself for analysis, making sure that your question is correct, making sure that you pick the variables correctly, and that you are collecting data in the way you need to, we're going to jump into quantitative. And again, Qualitative is important, and I definitely encourage people to explore it. I happen to be a quantitative person primarily, so that's what I'm going to talk about today, but don't ignore qualitative, so written feedback, interviews, that sort of thing. That is very important data, especially for things that can't be easily quantified. So we're going to revisit my example, and we're going to talk about how I would analyze it. So one thing that I'm going to do before I jump into my description is I'm going to share with you guys a Google folder in my Google Drive. And it has got the data that I use, the fake data that I use for this plant light study, the study data, as well as a Word document with a bunch of like the resources and links that you'll see further on in here, as well as a data set called the IRIS data set, which has been used in research, biological research for a really long time. It is really fun to play with, and there's lots of studies out there that have used it. So that's something that you can revisit and play with and do a lot of this stuff with. And I'll go through as we go on what is in those different files. But just in case you wanted to kind of follow along with the plant light data as we get going, I wanted to make it available. So if you have difficulty accessing it or you have questions about it, do drop those in the chat, let me know. But generally, so starting with data analysis, the first thing that you should do is organize your data, right? Like get everything set up. And I use Excel for everything. So I don't know if you all want to raise a hand or give a reaction if you have access to Microsoft Excel. Okay, there's a couple thumbs up. Awesome. So a lot of what I'm talking about today is in Excel. So if you all haven't used it or might have access to it and don't know, you might want to just kind of make note of things. And I'll drop these when we're done. I'll drop these slides also into this folder. So if you keep it open and refresh in about an hour, it'll show up. But a lot of this is in Excel. There are other open source Excel or spreadsheet files or apps that will do similar things, but it won't be quite the same. So just so you know. In any case, so usually I set up my data in this way, and you can see if you glance at the IRIS data set that they've done something similar. So I downloaded that data set exactly as is. You have different variables that you pick to go along the top. In my case, because I was looking at, in this example, looking at speed over time, I have each day. But you can set it up in whatever way you need to. But it allows you to look at your data overall, kind of get an idea about, okay, consistency, like I can check out errors, I can see different formats, I can see each of the individuals. So you can look across a row and say, like in row 2 with red light plant number 1, on day 35, it was shorter than day 20. Well, that's probably not right. So I can go back and check my source data. So the first thing that I do with any data set, and which I encourage you all to do, is to organize it in a spreadsheet and look it over just for obvious flaws. It's a lot easier to do with a small data set like this, but and I'll show you as we go through, there are ways to do it with larger data sets as well. So one other thing that I do that you'll notice that they do in the IRIS data set, if you look at that, is that they have it separated out by one of the independent variables in that case is the species of iris. And it's helpful to have that in its own category, and I will show you why. So the next thing you want to do is descriptive statistics. And this is where if you have a large data set, it's easier to, instead of looking over hundreds and hundreds and hundreds of rows of data, it might be easier just to jump to descriptive statistics and say, okay, what's here? Because you'll start to see in the range, like if you're supposed to have only measurements that start at 5 and you've got something that's a -2, you can say, okay, well, something's not right in this data, and it will help you narrow in. It helps you describe what's happened, and that's usually the first things people want to know. And it helps you really understand the nature of what you've collected. So looking at these, I'm sure most of you all are familiar with kind of the main ways that we summarize data. And I've got, central tendency, which is big and depends on the kinds of data that you have. Mean is often what we go with, but sometimes you want to have a mode. It makes more sense. So in some of that survey data where you've got different levels of responses that are, which option do you prefer? What kind of pizza do you like? you probably want to do mode. It might make more sense than doing an average, because how do you average nominal data? So this is when I was talking about scale, where that comes into the analysis piece. And it's important to think about the fact that distribution in your data makes a big difference, that in normally distributed data, the mean, median, mode will be the same, assuming that your data can be put in this format. You can't do this with like ordinal data, as I mentioned. But with other kinds of data, if you have a skew, then they'll be separate. And I'm not going to belabor the different descriptive data analyses, but it's definitely something to think about as you're selecting, okay, what makes the most sense for this data set? If I have outliers that are pulling my mean one way or the other, maybe I want to look at other measures of central tendency as well. So the next step is, as part of this, looking at the descriptives, aggregating your data in a format that is helpful, like means, is usually the next thing I do. And including the sample size is really important, because sometimes we forget, okay, what was the original data? So when you do the aggregates, make sure that you do that. Pivot tables. How many of you all have used pivot tables before? Thumbs up, anyone who has used them. Ah, yes, Miss Sarah, thank you. I'm glad to see that others have joined for a long time, I thought pivot tables seemed kind of complicated, and they have made a huge difference. I know that there is value in learning how to calculate things like averages and standard deviations by hand, but if you need to aggregate things and you're past the point of needing to understand the math underlying it, pivot tables are one of the best ways to explore data, and it's usually how I start. And with large data sets, it can be really, really helpful, because it can give you a lot of information very quickly. So like I said, range. So in this case, I just made my aggregate table using the average for each day, the average height for each day in centimeters. But you can find all kinds of things. You can do sums, you can do ranges, you could do basically any aggregate information that you need. But this is where it's helpful to have that light color column to differentiate. So those of you who have used pivot tables, I would love for you to drop in the chat kind of how you use them and what you use them for in the past. But I don't want to put anyone on the spot. So just if you'd like to drop it in there, I'm sure everyone else who's attending would find it helpful, but don't feel obligated to. So after you've done your basic kind of data review, you've organized your data and you've taken a look at it, and you've aggregated everything into a way that makes sense, whether you've gotten means or you've gotten sums or you've gotten medians, whatever it is that you're looking at. Visualization is the next way to explore the data. And for a lot of students, especially younger students, this might be where their data analysis ends, right? Like it's helpful to understand the data and to draw conclusions from it using visualizations. But they might not move on to the next step. So doing inferential statistics, this might be all they need. So in my example, going back to the lights, a line graph was the first approach that I took. And it's useful because by connecting all the different points, it gives you a real sense of growth, right? It gives you change over time, which is what I was looking for. I wanted to see, how does this change over time? What I'd like to point out, and what you can see on here, is that because of the way I set it up in the spreadsheet, you can see that the data points look like they're evenly spaced, even though day 5 and day 7, look like it's the same distance as day 1 and day 5. It's something to really be aware of, because that's not an accurate representation of the data. You want to be really careful with thinking about how your data is presented in order to draw conclusions, but also so that your reader knows what's going on. So this, you can see in the original where they're evenly spaced, and then in the updated where you can actually see the differences between days in a reasonable in the more accurate format. So line graphs are really great for showing individual, or in this case, aggregate change by group over time. It's honestly one of the best ways to see any change that you have multiple time points for. And by multiple, 3 is the absolute minimum, though that doesn't usually tell you too much. I usually would look for, 5 or more to get an idea of what, to get a real idea of the pattern or the rate. Histograms are another great way of visualizing your data. They look like bar charts, but they're slightly different in that these are bins. So this is actually using the iris data. And you can see, so petal length at the end, instead of using it as a bar chart by any other category that they could use, it's everything that's between, 1 and 1.5. It's the number, the frequency of occurrences. So it kind of gives you an idea overall of what the distribution looks like. It really depends on the kind of data that you have, whether or not you want to use a histogram, but it can be really useful to look at and see, okay, well, it looks like we've got a bunch that end up between 1 and 2, so that's interesting. And then others that there's this dip between 2 and 3. You can ask interesting questions and explore in interesting ways. So then the bar chart, which is kind of like the typical, like what you think of when you see something along those lines, which in this case, this is using the light data and it's showing height. It's easier with large changes to look at this, whereas probably line graphs are better with the small changes. I strongly recommend that you use 0 at the beginning of your x-axis unless there's a real reason not to. Otherwise, it gets very confusing. And then this goes back to your kind of data, right? If there's not a true 0, then having 0 on that axis isn't necessarily helpful. I also recommend horizontal bars if you have a lot of categories, because it's easier to compare. It's just your eye is drawn, because the way that we read from left to right, your eye is drawn out and it gives you a better idea. The colors also really help. So whether you're comparing groups or whether you are just looking at, in this case, chunks of different categories, it's really helpful to add color, add shading, usually trying to make sure at least that it's easy to view. So if you can't look at a figure and in the first 30 seconds have an idea of what they're talking about, even if you took away all the labels, then you probably want to update your visualization. Scatter charts are really good for looking at relationships. And actually in the iris data set, I have done one and I didn't label species or anything. I just did a scatter chart looking at both the, it's only for the petal length and width. And you could see where they kind of group together, and you could almost identify which species is which just based on where they group together. So in a larger data set, it can give you really intriguing information. You can see with the smaller data set, though, it's really hard to pinpoint what am I looking at here with these individual dots. Now, these are aggregate points and they're not each individual plant. So it might be that we would see more with individual plants. And so usually with scatter charts, unless there's just a ton of data, I usually recommend going ahead and using the individuals, because it gives you an idea of the dispersion of those points as well. But otherwise, it's probably easier just to use like a line in between. So in this example, that the authors actually used averages, but they drew lines so that you have an idea of what the pattern is that they're seeing. So one is a direct linear relationship line, and the other they kind of curve with the data, but either way you can see that there's a downward slope. And so if you want to use averages and you want to include a line, I would recommend making sure that the line is meaningful and deciding why you would include it. Pie charts we use a lot. We really enjoy them for certain categories of things, usually for nominal data like this by country, but it's really hard to read with multiple categories. Like I can't glance at this and know exactly what I'm looking at. It's a lot of glancing back and forth. So a bar chart probably would be better. And in general, rules of thumb with visualizations. Visualizations are so helpful and they're one of the best ways to present your data, but make sure that your colors are consistent. So like with mine, I had always the red light was in a red line or red dots. I didn't change it. Make sure that your numbers have the same number of decimals, that everything looks really consistent and easy to interpret for your reader. As I mentioned before, the essential features should be interpretable on their own. Like within 30 seconds, you should have an idea of kind of what's going on in the visualization. And once again, about misrepresentation of your axes and spacing, you want to make sure that your data genuinely is genuinely represented within your visualizations. So you don't want to make it seem like, oh, it's a linear kind of process, when actually, if you had the spacing correct, it ends up kind of peaking and petering out. So those are kind of rules of thumb with visualization. This is a chart that is really, really helpful, which will also be in the Google Drive when all is done. But it gives you an idea of the kinds of visualizations that you might consider for your data and for your kind of analysis. So when you're thinking about like a relationship, as I mentioned with the scatter chart, it's really easy with 2 variables. With 3 variables, there's something called a bubble chart, which you might have seen, where the bubbles actually gain in size depending on the number of people that are present. So this is a really helpful start. But if you put your data in a format and you're like, this doesn't tell the whole story, you can pick a different format. I think it's just really useful to visualize data in multiple ways before you settle on one. So we're going to jump a little bit into inferential statistics. We're not going to go super in depth because, again, that's could be, it's entire master's level course on just getting into the introductory. I would encourage those of you, so how many of you guys are at the high school level, either as a student or a teacher or a parent at the high school level? All right, got a couple hands going up. Awesome, cool. So especially for high school students, this should be pretty, at least the ones that I'm going to go through today are pretty standard, pretty straightforward. You all probably used to it. I strongly recommend that those of y'all at the lower levels, if you can, introduce some of these, especially to your students who are a little bit more advanced, because they're not overly complicated. And I think that they really help students get a step in to understanding what we're doing in inferential statistics. Things like t-tests, you can actually do by hand. Not that they need to, but it's helpful to see how data analysis happens. So a reminder that inferential statistics, again, sample size is really important because the idea from them is to make inferences about a population from which you've sampled. So it's not generalizable necessarily if you have a sample that's not representative or is too small from your population. So as I mentioned, t-tests, they look between differences in 2 groups. So this is really nice for something like if you had the light study that I looked at, you only had 2 colors, or you had one in sunlight and one in like a red light or something like that, then you could see, okay, how tall did the plants grow? And can I see a statistical difference between them? And then correlations are another one, which I'm sure most of you all are familiar with, looking at the association between variables. And this is a fun one to look at, like with the iris data, because there is a correlation between the petal length and petal width and the sepal length and sepal width. So it's looking at trends. You have to usually remind students that correlation does not equal causation, right? Like these things vary at the same time, but that does not mean that one makes the other happen. So just something to keep in mind as you use these. The ones who do use this, if you don't have statistical software that you use on a regular basis, GraphPad has great quick calc is that you can run it in your browser and you can use it with all the different kinds of data that we talked about. So categorical data, we talked about, like the nominal level scale, continuous data, which is your interval and your ratio data. There's all kinds of analyses that they have, and it's pretty easy to use. And again, this is in the resources folder, and the link is in there. So don't feel out, you have to scribble all this down, and the slides will be in there as well. Other resources that I strongly recommend that I have used before and that I think are helpful. NCES Kids. So the National Center for Education Statistics has great data. And so if you're looking for a data set, so especially for students who might not have the time or ability to run their own experiment but want to explore data, they have data sets that you can pull variables from and do their own research on the data set. It is fantastic on almost any kind of educational data that you have interest in, from early childhood to teacher level data. And they have this Kids Zone, which is specifically for the younger students, but provides an opportunity to, build a graph for students who might not have access to. That is an interesting point. Yes, so in general, so there's a question in the chat. Technically, aren't you only allowed to use t-tests if your data is normal, not just continuous? And yes, so if you think about the distribution that we had before where your heavily skewed data is over to one side, t-tests are made for normal data, and there's a number of other assumptions that go with them, especially paired t-tests. But if you have a sufficient sample size, in general, t-tests are pretty robust to violations of non-normality, unless your data is just hugely skewed, which in general, if you have enough, a large enough sample size, they should be okay. And that's the other side of it also. Sometimes you can have really quirky data, but because your sample size is so large, your t-test will be significant, even if practically there's not an actual difference between the groups. So it really is important to think about your sample and your data that you have before diving in. But yes, that is an excellent point. But okay, so sorry, back to NCES Kids. It allows students to make graphs if they need to, especially if they don't have access to Excel. Question in the chat is, would NCES Kids be useful for those who have had little to no exposure to graphs or data analysis, even at the middle school level? Yes, so there were actually tutorials within NCES Kids that give some background. There's resources for teachers, and also like little quizzes. So the test your knowledge tab on the side can let students kind of get an understanding of what they're actually doing with the data before they jump into the graphic part. They've got a lot of resources there. So it's a really excellent resource. Depict Data Studio is another visualization. So this is probably for more advanced users who have a better idea of what they're looking for. And their data, they can say, okay, I'm measuring 2 time points and I've got this kind of data and I'm going to do that. So it shows you all the different kinds of approaches that you might want to take. So in this case, I selected 2 points in time. And so it's got a bunch of different visualizations that might be helpful. So while I might have thought, oh, I've got 2 points in time, I want to do something with the line. Deviation graph might be a really good way to go, or a clustered bar. So it gives you other ideas for really informative visualizations. This is usually for teachers. Some more advanced students might use it, but the American Statistical Association has education resources that are really helpful for getting students started with statistics, helping them understand what they're looking at and interpreting graphs. So getting a little bit past the analysis into the interpretation piece. And I highly recommend exploring those resources as well. And then of course there's a crash course that's on YouTube where they talk through all of this stuff in much more detail for how to actually do a lot of these stats. And it's a really great video, but it is 45 minutes long. Before I jump to questions, I'm going to stop share quickly, and I am going to share, I want to show you all the NCS website since it was mentioned. Okay, can you all see my screen again? Okay, so NCS Kids, this is where they have education quiz, math teachers, they have a lot of different materials. Educators Corner has a lot of information for teachers that's really helpful. I also recommend, especially for older students who have a little bit of familiarity with stats, these are the data sets in their data lab. You can go to all these different data sets and you can use, so let's say we want to create a graph looking at different data. So you can drag different variables over and you can create tables just based on data that's already there. So it gives if you want students to have an opportunity to explore data and maybe not necessarily do a lot with it, maybe they're familiar with graphs, they just really want to dive into data, this is a really great resource. GraphPad is one that I mentioned earlier, and it's got all of these different measures and approaches. They don't do a great job with actually teaching. I mean, they have a little bit of information and you have to go through and read the examples. So I wouldn't say it's a standalone resource where you could just drop students into it, but it will serve as a reminder, and then they can enter their data and calculate from there. And then for the folder that I shared with you all, if you go through and you look at, you can see all the different tabs that I've created. So everything that I've talked through is here. The pivot table, if you download this to Excel, it has the pivot table totally set up so you can recreate this. It's got the line graphs, and then the line graphs with the, well, the line disappeared, but with the appropriate, with the spacing. Somehow that ended up being the scatter plot. I'll correct that. And then a bar chart. So for those of you who are maybe less familiar with Excel, you can use this to kind of play with, copy and paste, see how things work. There's a question. Does your drive have the links? Yes. So in the drive, there is a resources and links document. So it's got all the links in here, the sample size, including this table that's from sciencebuddies.org, which I really like because it's a great, it gives you a great kind of like concrete information about like, okay, why sample size matters, like how the margin of error increases as a sample size decreases. So if you only have 10, it's got, you know, margin of error of like 31.6%. So I happen to find it really helpful for visualizing, but also all of these other links are on here as well. And then the IRIS data, as I mentioned, is a data set that has a long history. So you can read up on it in Wikipedia, and then it's got all of the, it's just pulled it off of here. It's really easy to dive in and work with. And I strongly recommend that you guys, if you have any interest in just kind of playing with data, I pulled out the pedals and did a pivot table just from it. It gives you an opportunity to jump in and do that. So, oh, you're welcome. So that is it was kind of a quick run through. I know it's a lot of information. Would love to answer any questions that you guys have. So we've got a little bit of time left. I could also walk through any of the data that you would like to see in more detail as well. But I wanted to make sure that we had time for some questions, and specifically about your own science fair struggles or questions with data that you might have. So please feel free to speak up or drop questions in the chat, and I am more than happy to answer them. I will also drop my email in the chat. So if you say to yourself, I don't want to necessarily ask my question right now, or my question hasn't come to me, I really want to revisit the slides first, feel free to shoot me an email and ask any questions that you might have. Okay, so comment in the chat. I have a background in stats, but I find the biggest challenge is catching the kids early on collecting the right data. Yes, and so that's why I spent the first, half of this going through the kind of from questions all the way through to analysis, because what I find is that if you don't have the right data, there's no analysis that you could do at the very end that's going to make up for that. So really, really emphasizing, what's your question? What are the variables? What do you need to know? How will you know when you have your answer? And going through that process. When I worked with middle school students, that's always where I start. We spend a lot more time on that than we do even getting to the analysis point, because I find that with a lot of them who are doing descriptives or really like intro stats, the biggest thing is understanding what data they need to collect and the attributes of their data as they're getting started. What can be the easiest stats website for middle school students to use? That is a great question. So I am not going to, the ones that I've recommended I really enjoy, but I won't necessarily advocate for any one site depending on what the needs of your students are. So I guess it depends on if they need like background to learn from, if they need data to play with. Different, I think it depends on the level of the student and where they are and what they're looking for. So I apologize, that's not a concrete answer, but it really is dependent on the student population and what you're looking for them. If you want to provide a little bit more info. Yeah, scientific method is super critical. And it is very much, I find that often, especially starting the elementary level, we treat science like it's a recipe. Like you do X, you do Y, and then boom, Z will happen. And that is not the way that scientists do science. Like the work of scientists is rarely that linear. So really also talking about kind of some of the uncertainty, like what do you do when you get to results that don't make any sense? Is it the data that you've collected? Is the way you've analyzed it? Or is there something else that you didn't measure? And so really getting at all the different, you know, bits of nuance that go into science can be really complicated. Yeah. A lot of high school kids still struggle with this. Find high schoolers want to collect a lot of categorical data and analyze it all together. Yes, yes. And that is, you know, also why I started with scales of data and what you're planning to do with data, and really talking through, you know, what is the purpose of what you're trying to get at, and really making them operationalize their variables and talk about like, okay, what do you mean by this? And what are you looking for? Like the precision is so important to make sure that you are getting at the right data, because again, the analysis, and it's not even just high school students. I have had faculty come to me with the survey and say, I've got this survey data, will you help me analyze it? And I'll say, yeah, absolutely. And I look at it and I'm like, you don't have the data that you need to answer the question that you want. We can answer other questions, but it's not going to be the question that you're looking for. And even things like with surveys, you know, people will say, well, I want to know, you know, if my class is good. And I'm like, well, what does that mean? What do you mean by good? You have to define it. So it takes a lot of that like back and forth question answering, like, so what does that look like? How do you measure it? What do your measures look like? Or do you have a measure that is ranking things versus measure, like what numbers are you looking at? And it really can be a struggle to wrap your mind around going into it. So yes, the struggle is for a lot of folks. Yes, and so I saw the comment about taking AP statistics, and I will say that it's interesting because sometimes stats don't really sink in until you have to use them on something. Yeah, it's sometimes like using things. So I was working with a group of students who were, this was a couple of years back, but it was, I want to say like ninth grade. They're either 8th or 9th grade. And we used football statistics. So we downloaded like player stats, and we had, you know, made hypotheses. Like I feel like having students, and I think that's the other part of science fair projects, is having students really care about what they're looking at. If the curiosity is not there, then, you know, there's no, if my data's off, do I really care? I don't like pea plants anyway. So really thinking about like, okay, what is interesting? And that's why, you know, if you can get data sets that are collected from elsewhere that have something interesting in them. Like the NCES has great data, but I love education and I love statistics. So for me, it's fun to dig into. But there's a data sets in all kinds of areas that you can find for free all over the place that might have more interesting features that students might really jump into and be interested in using.
I think a lot of what you know, Ms. Curry, you're sharing there with high school and middle schoolers is true, right? So over the last few years with our science fair project, the judges are starting to tease out a lot of the decisions could be based solely on who did better statistics, not necessarily who did better statistics, but who used the right analysis tool for their data. And we've seen that quite a bit, where there's been a lot of development in students' projects and they're doing beautiful work. But when it comes to collecting the data or even analyzing what is the data saying, students tend to over extrapolate, for example. But it does come down to, all right, was this analysis method appropriate for this project? Is it appropriate for the questions they're asking? And so Dr. Newell here is trying to crunch a lot of what, you know, I shared with her into a 1-hour session that should really be parsed out into smaller sections where we can spend more time in that whole development. All right, this is the research question. What do we need to do to ensure that we're answering these questions? So Dr. Newell, thank you so much for trying your best to give us all that information. Teachers, students, parents, please take down her email, because as you go along, or if you have questions for your students, when they start to do their analysis part, this is a great resource at our fingertips here.
Yeah, absolutely right. It's difficult to analyze data. It's difficult for even, I mean, so my area of emphasis, like my statistical approaches that I'm really in on, I'm regression based. I'm in hierarchical linear models. So if someone comes to me with a statistical approach that I'm less familiar with, I have moments where I'm like, oh my gosh, okay, let me do some background reading. Let me like reintegrate myself into this. Um, Data analysis is complex. There's a lot of crimes against data that happen, even with people who should know better, where they take data that shouldn't be used for a purpose and use it in that purpose. So even little things like, I was talking about like the line graph, right? And the spacing, you'll see in like, it's not as common now, but like in a lot of the old like USA Today, I don't know if they still do, used to have a box at the bottom where they have like a little figure. And on some of those, it's like, what scale are you using with this? It looks like a huge difference, but if you plot it and you actually have a zero, then it's not. So even at that level, even before you get statistics, there's crimes against data that happen all the time. So really thinking about, what data needs to be collected, how it should be presented. And then once you get into the statistical analyses, okay, really diving into before you talk about, and I apologize because Ms. Gurry, you absolutely pointed out, it's necessary to understand the assumptions that go along with all these analyses as well. So when you teach students about using a t-test, there's more even than just normality of data. If you teach students about using an ANOVA, which is the next level up from a t-test, where you've got 3 groups, you're comparing 3 groups, 4 groups, 5 groups, what are the assumptions that go with that? Because if you use data that is not appropriate and you come to conclusions, your conclusions are going to be fundamentally flawed. And people do it all the time. We're like, well, this is easy. I'll just use this online program and I'll drop my data in and now I've got results and it's beautiful. And someone will say, well, wait, I'm looking at your data and I have questions about your distribution, or I have questions about how you measured this. So yeah, it's complex. It's not easy, but it's something that, especially as we're in an increasingly data-driven world, it is so important to start learning about and really immersing ourselves in. So.
Great. Well, again, please feel free to email Dr. Newell, and she's there at the Baylor College of Medicine. With that, we have, we're just about at lunch. So we have an open period for lunch this afternoon. Feel free to come back to this session at 12:40, and we'll get to speak to individuals who started science fair scholars on their campuses, and also meet and greet with some ISEF finalists. So again, thank you, Dr. Newell.